In this video, we will be presenting a new method of investigating GPCR signaling by employing recombinant receptors activated by inert synthetic ligands. G-protein coupled receptor, or GPCR, is one of the most important classes of signaling receptors and a subject of constant research. Unfortunately, it is difficult to study the physiological effect of GPCR signaling in vivo due to the presence of varying levels of endogenous ligands. Furthermore, traditional experimental techniques tend to cause systemic changes and hence are inadequate in assessing tissue or organ-specific signaling pathways. To combat these issues, DREADs, or designer receptors exclusively activated by designer drugs, are invented in 2007 by Armbruster et al. It is a class of genetically engineered recombinant GPCRs that are designed to be specifically activated by inert drug-like compounds. DREADs are not affected by endogenous ligands, thereby allowing investigators to control its spatial activation when they are transfected into specific tissues using viral vectors. Two components are necessary for this system to work: an inert ligand and a modified GPCR. The most commonly used ligand is clozapine N oxide, or CNO, since it is inert, highly potent, and has high bioavailability in host. Muscarinic receptors are typically chosen as the basal GPCR template. Site-directed mutagenesis is performed in order to abolish their activity for acetylcholine, the native ligand, while maintaining high affinity for CNO. This is accomplished by converting residue 33 on the third transmembrane domain, a tyrosine, to a cysteine, and residue 46 on the fifth transmembrane domain, an alanine, to a glycine, for all muscarinic receptor classes M1 through M5. With such modifications, CNO is demonstrated to have similar potency to M3 DREADs as acetylcholine to native M3 receptors, allowing DREADs to replicate the effect of native receptors when stimulated. If acetylcholine is added to M3 DREADs and CNO is added to native M3 receptors, severe potency reduction of greater than 40,000-fold can be seen in both cases. One common use of DREADs is to assess the effect of neuron silencing in the brain. M4 GPCRs, which are coupled to GI, are known to induce the opening of potassium channels when activated by acetylcholine, resulting in neuron hyperpolarization and inhibition of neuron firing. Recombinant M4 DREADs, or HM4D, are shown to be just as effective in inducing the silencing of hippocampal neurons when activated by CNO, which is evidenced by the voltage traces of cultured hippocampal neurons. With the addition of carbachol, an acetylcholine analog, hippocampal neuron control group experiences hyperpolarization, represented by the dip in the voltage trace, whereas CNO did not have any effect. Similarly, in neurons expressing HM4D receptors, CNO addition resulted in hyperpolarization, replicating carbachol's effect on native M4. Furthermore, the expression of HM4D does not affect native M4 function or baseline neuron activity, since carbachol can still induce hyperpolarization in HM4D expressing neuron population by targeting native M4. DREADs have now been used extensively to elucidate neuronal pathways in specific areas of the brain thanks to their ability to selectively modulate neuron activity. For example, DREADs have played an important role in Down syndrome research, where selective activation of HM3DQ receptor by CNO in the locus coeruleus of the brain has shown to improve memory. This finding stimulated directed research effort in search of a therapeutic agent that can selectively target locus coeruleus M3. On the other hand, there are also some limitations to utilizing DREADs. Firstly, they're not optimal for experiments that require precise temporal control. CNO acts on the order of minutes, while optical methods of neuronal activation can act on the order of milliseconds. Additionally, it is difficult to control the level and duration of neuronal stimulation produced by DREADs. Hence, next-generation DREADs will focus on resolving these issues by searching for a more optimal inert ligand while improving GPCR design to offer better control. Despite these limitations, the current DREAD technology remains a very promising novel method of investigating neurophysiology.